Hello students, let me talk about antigen-antibody interactions. Introduction. The antigen-antibody interaction is a bimolecular association similar to an enzyme-substrate interaction, with an important distinction. It does not lead to any irreversible chemical alteration in either the antibody or the antigen. The association between an antibody and an antigen involves various non-covalent interactions between the antigenic determinant or epitope of the antigen and the variable region VHVL domain of the antibody molecule, particularly the hypervariable regions or the complementarity determining regions or CDRs. The exquisite specificity of antigen-antibody interactions has led to the development of a variety of immunologic assays which can be used to detect the presence of either antibody or antigen. Immunoassays have played a vital role in diagnosing diseases, monitoring the level of the humoral immune response, and identifying molecules of biological or medical interest. Strength of antigen-antibody interactions. The non-covalent interaction that forms the basis of antigen-antibody binding include hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. Because these interactions are individually weak, a large number of such interactions are required to form a strong antigen-antibody interaction. The combined strength of the non-covalent interactions between a single antigen binding site on an antibody and a single epitope is the affinity of the antibody for that epitope. Low affinity antibodies bind antigen weakly and tend to dissociate readily, whereas high affinity antibodies bind antigen more tightly and remain bound longer. The association between a binding site on an antibody with a monovalent antigen can be described by the equation where K1 is the forward association rate constant and K-1 is the reverse or dissociation rate constant. The ratio K1 by K-1 is the association constant Ka, a measure of affinity. Because Ka is the equilibrium constant for the above reaction, it can be calculated from the ratio of the molar concentration of bound antigen-antibody complex to the molar concentration of unbound antigen and antibody at equilibrium as follows. Ka equal to concentration of antibody-antigen complex divided by antibody concentration into antigen concentration. The value of Ka varies for different antigen-antibody complexes and depends upon both K1, which is expressed in units of liters per mole per second, and K-1, which is expressed in units of liters per second. For small haptens, the forward rate constant can be extremely high. In some cases, K1 can be as high as 4 × 10⁸ liters per mole per second. For larger proteins antigens, however, K1 is smaller with values in the range of 10⁵ liters per mole per second. The rate at which bound antigen leaves an antibody's binding site, that is, the dissociation rate constant K-1, plays a major role in determining the antibody's affinity for an antigen. For example, the K1 for the DNP L-lysine system is about 1/5 that for the fluorescence system, but its K-1 is 200 times greater. Consequently, the affinity of the anti-fluorescent antibody Ka for the fluorescence system is about 1000-fold higher than that of anti-DNP antibody. Low affinity antigen-antibody complexes have Ka value between 10 raised to 4 and 10 raised to 5 liters per mole. High affinity complexes can have Ka values as high as 10 raised to 11 liters per mole. Avidity. The affinity at one binding site does not always reflect the true strength of the antibody-antigen interaction. When complex antigens containing multiple repeating antigenic determinants are mixed with antibodies containing multiple binding sites, the interaction of an antibody molecule with an antigen molecule at one site will increase the probability of reaction between those two molecules at the second site. The strength of such multiple interactions between a multivalent antibody and antigen is called the avidity. The avidity of an antibody is a better measure of its binding capacity with biological systems. For example, the reaction of an antibody with antigenic determinants on a virus or a bacterial cell, then the affinity of its individual binding sites. High avidity can compensate for low affinity. For example, secreted pentameric IgM often have a low affinity than IgG, but the high avidity of IgM resulted from its high valencies enable it to bind antigen effectively. Cross interactions. Although antigen-antibody interactions are highly specific, in some cases, antibody elicited by one antigen can cross-react with an unrelated antigen. Such cross-reactivity occurs if two different antigens share an identical or even similar epitope. In the latter case, the antibody's affinity for the cross-reacting epitope is usually less than that for the original epitope. Cross-reactivity is often observed among polysaccharide antigens that contain similar oligosaccharide residues. A number of viruses and bacteria have antigenic determinants identical or similar to normal host cell components. In some cases, these microbial antigens have been shown to elicit antibody that cross-react with the host cell components, resulting in tissue-damaging autoimmune reaction. The bacterium Streptococcus pyogenes, for example, expresses cell wall protein called M antigens. Antibodies produced to streptococcal M antigens have been shown to cross-react with several myocardial and skeletal muscle proteins and have been implicated in heart and kidney damage following streptococcal infections. Precipitation reactions. Antibody and soluble antigens interacting in aqueous solution form a lattice that eventually develops into a visible precipitate. Antibodies that aggregate soluble antigens are called precipitins. Although formation of the soluble antigen-antibody complex occurs within minutes, formation of the visible precipitate occurs more slowly and often takes a day or two to reach completion. Formation of antigen-antibody lattice depends on the valency of both the antibody and antigen. The antibody must be bivalent. A precipitate will not form with monovalent Fab fragments. The antigen must be either bivalent or polyvalent. That is, it must have at least two copies of the same epitope or have different epitopes that react with different antibodies present in the polyclonal antisera. Precipitation reaction in fluids yield a precipitin curve. A quantitative precipitation reaction can be performed by placing a constant amount of antibody in a series of tubes and adding increasing amount of antigens to the tubes. At one time, this method was used to measure the amount of antigen or antibody present in a sample of interest. After the precipitate forms, each tube is centrifuged to pellet the precipitate. The supernatant is poured out and the amount of precipitate is measured. Plotting the amount of precipitate against increasing antigen concentrations yield a precipitin curve. Excess of either antibody or antigen interferes with maximum precipitation, which occurs in the so-called equivalence zone, within which the ratio of antibody to antigen is optimal. Precipitation reaction in gels yield visible precipitin lines. Immune precipitates can form not only in solution but also in an agar matrix. When antigen and antibody diffuse towards one another in agar, or when antibody is incorporated into the agar and antigen diffuses into the antibody containing matrix, a visible line of precipitation will form. As in a precipitation reaction in fluid, visible precipitation occurs in the region of equivalence, whereas no visible precipitate forms in the region of antibody or antigen excess. Two types of immunodiffusion reactions can be used to determine relative concentration of antibody or antigens, to compare antigens, or to determine the relative purity of an antigen preparation. They are radial immunodiffusion, the Mancini method, and double immunodiffusion, the Ochterlony method. Both are carried out in a semi-solid medium such as agar. In radial immunodiffusion, an antigen sample is placed in a well and allowed to diffuse into agar containing suitable dilution of an antiserum. As the antigen diffuses into the agar, the region of equivalence is established and a ring of precipitation, a precipitin ring, formed around the well. The area of the precipitin ring is proportional to the concentration of the antigen. By comparing the area of the precipitin ring with a standard curve obtained by measuring the precipitin area of known concentrations of the antigen, the concentration of the antigen sample can be determined. In the Ochterlony method, both antigen and antibody diffuses radially from wells towards each other, thereby establishing concentration gradient. As equivalence is reached, a visible line of precipitation, a precipitin line, forms. Agglutination reactions. The interaction between antibody and a particulate antigen results in visible clumping called agglutination. Antibodies that produce such reactions are called agglutinins. Agglutination reactions are similar in principle to precipitation reactions. They depend on the cross-linking of polyvalent antigens. Just as an excess of antibody inhibits precipitation reactions, such excess can also inhibit agglutination reactions. This inhibition is called the Prozon effect. Several mechanisms can cause the Prozon effect. First, at high antibody concentrations, the number of antibody binding sites may greatly exceed the number of epitopes. As a result, most antibodies bind antigen only univalently instead of multivalently. Antibodies that bind univalently cannot cross-link one antigen to another. Prozon effects are readily diagnosed by performing the assay at a variety of antibody or antigen concentrations. When one is using polyclonal antibodies, the Prozon effect can also occur for another reason. The antiserum may contain high concentrations of antibodies that bind to the antigen but do not induce agglutination. These antibodies, called incomplete antibodies, are often of the IgG class. At high concentrations of IgG, incomplete antibodies may occupy most of the antigenic sites, thus blocking access of IgM, which is a good agglutinin. The lack of agglutinating activity of an incomplete antibody may be due to restricted flexibility in the hinge region, making it difficult for the antibody to assume the required angle for optimum cross-linking of epitopes on two or more particulate antigens. Alternatively, the density of epitope distribution or the location of some epitopes in deep pockets of a particulate antigen may make it difficult for the antibodies specific for this epitope to agglutinate certain particulate antigens. When feasible, the solution to both of these problems is to try different antibodies that may react with other epitopes of the antigen that do not present these limitations. Agglutination reactions are routinely performed to type red blood cells or RBCs. In typing for the ABO antigens, RBCs are mixed on a slide with antisera to the A or the B blood group antigens. If the antigen is present on the cells, they agglutinate, forming a visible clump on the slide. Determination of which antigens are present on the donor or recipient RBCs is the basis for matching blood type for transfusion. Many immunoassays have been developed on the basis of antigen-antibody interactions that have vast applications in research and medical diagnostics. Some of them are radioimmunoassay, a highly sensitive and quantitative procedure that utilizes radioactively labeled antigen or antibody. The enzyme-linked immunosorbent assay or ELISA that depends on an enzyme substrate reaction that generates a colored or chemiluminescence reaction product. Western blotting. In Western blotting, a protein mixture is separated by electrophoresis. Then the protein bands are electrophoretically transferred into nitrocellulose papers and identified with labeled antibody. Fluorescent microscopy. It uses antibodies labeled with fluorescent molecule to visualize antigen on or within cells. Flow cytometry. It provides an unusually powerful technology for the quantitative analysis and sorting of cell population labeled with one or more fluorescent antibodies. Conclusion. In conclusion, I would like to say that antigen-antibody interactions depend on weak interactions. The affinity constant provides a quantitative measure of the strength of the interaction between an epitope of the antigen and a single binding site of an antibody. The interaction of a soluble antigen and precipitating antibody in a liquid or gel medium forms an antigen-antibody precipitate. The interaction between a particulate antigen and agglutinating antibody produces visible clumping or agglutination that forms a basis of simple, rapid, and sensitive immunoassays. There are many immunoassays that have been developed on the basis of these antigen-antibody interactions, and it has vast application in scientific research and medical diagnostics.